above 200 nanometer. Components of UV visible spectrophotometer: source, filters and monochromators, sample compartment and detector and recorder. Source: a stable source of radiant energy at the desired wavelength. Wavelength selector: a device that isolates a restricted region of the EM spectrum used for measurement. Monochromators, prisms and filters. Sample container: a transparent container used as hold the sample, cells and cuvettes, etc. Detector and photoelectric transducer: converts the radiant energy into signal. And signal processor and readout: amplifies or attenuates the transduced signal and sends it to readout devices as a meter, digital readout, chart recorder and computer, etc. UV instrumentation. Various UV radiation sources are as follows: deuterium lamp and hydrogen lamp, tungsten lamp, xenon discharge lamp, mercury arc lamp. Various visible radiation sources are as follow: tungsten lamp, mercury vapor lamp and corbonan lamp. Wavelength selector's output a limiter, narrow, continuous group of wavelengths called a band. Two types of wavelength selectors. First one, filter, and another one, monochromator. Filter: two types of filter. There are interference filters and absorption filters. And then monochromators: wavelength selector that can be continuously scan a broad range wavelengths, used in most scanning spectrometers including UV visible and IR instruments. Prism type, refractive type and reflective type, and grating type, diffraction type and transmission type. Sample compartment. Spectroscopy requires all materials in the beam path other than the analyte should be as transparent to the radiation as possible. The geometries of all components in the system should be such as maximize the signal and minimize the scatter light. The material from which a sample cuvette is fabricated controls the optical window that can be used. Some typical materials are optical glass 335 to 2500 nanometer, special optical glass 320 to 2500 nanometer, quartz infrared 220 to 3800 nanometer, quartz far UV ultravisible 170 to 2700 nanometer. And detectors. After the light has passed through the sample, we want to be able to detect and measure the resulting light. These types of detectors come in the form of transducers that are able to take energy from light and convert into an electrical signal that can be recorded and, if necessary, amplified. Three common types of detectors are used: barrier cells and photomultiplier and photoemissive cells detector. Detection impurities. It is one of the best methods for determination of impurities in organic molecules. Additional peaks can be observed due to impurities in the sample and it can be compared with that of standard raw material. By also measuring the absorbance of light or specific wavelength, the impurities can be detected. UV spectra of paracetamol. UV application. UV spectroscopy is useful in the structure elucidation of organic molecules, the presence of or absence of unsaturation, the presence of heteroatoms. And then The location of peaks and combination of peaks, it can be concluded that whether the compound is saturated or unsaturated, heteroatoms are present or not, etc. Quantitative analysis. UV absorption spectroscopy can be used for the quantitative determination of compounds that absorb UV radiation. This determination is based on Beers-Lambert's law, which is follows A equal to log I naught by I naught divided by IT, where epsilon is extinction coefficient and C is concentration and B is length of the cell that is used in UV spectrophotometer. And qualitative analysis. UV absorption spectroscopy can characterize those type of compounds which absorb UV radiation. Identification is done by comparing the absorption spectrum with the spectra of known compounds. Chemical kinetics. Kinetics of reaction can also studied using UV spectroscopy. The UV radiation is passed through the reaction cell and the absorbance changes can be observed. Detection of functional group. This technique is used to detect the presence or absence of functional group in the compound. And absence of band at particular wavelength regarded as evidence for absence of particular group. Quantitative analysis of pharmaceutical substances. Many drugs are either in the form of raw material or in the form of formulation. They can be assayed by making suitable solution of the drug in the solvent and measuring the absorbance at specific wavelength. Thank you.